recording and introduce our speaker today. Today, our presenter is Dr. Sharon Yeatts. Dr. Yeatts is professor of biostatistics at the Medical University of South Carolina, Department of Public Health Sciences, and the MUSC Data Coordinating Unit. She is the co-PI of the Siren Network DCC and the statistical PI for the IceCap and Boost 3 trials within Siren, as well as the Saturn trial within StrokeNet. So lots of great network experience. Today, Sharon will present her talk, which is titled What to Know Before Talking to Your Statistician About Sample Size. And her talk is an annual favorite among our CTMC webinar series. So we are so grateful to have her here again with us. So with that, I will turn it over to you, Sharon. Thank you.
Thanks, Megan. Once I start sharing, I think I probably won't be able to actually see the chat. So if you have a question, if it's okay with Megan, if you have a question in the chat that I haven't acknowledged and you think it's better to address it right then, please go ahead and feel free to interrupt me. I'm going to start by sharing a video that I share at the start of every one of these talks. So I apologize if you've already heard it. You'll just pretend to laugh again.
I have a study, and I need to know how many patients I need. I think I only need 3 patients.
Okay, what kind of study is it?
I'm doing a lab study. Can I just use 3 patients?
That depends. What are you trying to show?
We always use 3 patients.
Okay, what are you trying to show?
I'm trying to show that drug A and drug B are better than only drug A.
How are you measuring if it is better?
We are looking at apoptosis. Can we just use 3 patients?
What kind of measure of apoptosis? Do you know how variable that measure is?
We always use 3 patients. That is what we have published before.
3 patients per group or in total?
Yes.
Okay, let's try something different. What type of measure are you using? And is this an in vitro study? An in vivo mouse study?
It is an in vitro study of patient samples, and we are looking at measures of apoptosis after treatment with drug A and drug B versus drug A and control. And drug B alone.
Oh, how many groups are you analyzing altogether then?
My grant is due tomorrow. I just need to know if I can just use 3 patients per group.
Okay, well, you probably should have come to me earlier for help with this.
All I need you to do is tell me that 3 patients are okay.
So, it is not that easy. Your marker for apoptosis may have a large amount of variability associated with it that could make it difficult to ascertain differences based only on 3 patients per group. It also depends on what kind of outcomes you are looking at. Are you looking at normalized changes in expression levels from baseline? 3 patients may not be sufficient to detect differences in these expression levels between the different treatment groups.
I do not understand.
I'm saying that you may need more than 3 patients per group, and I need more information to determine that.
The serine protease granzyme D is made in cytotoxic lymphocytes, where it is inactive due to the low pH of the storage granules. It is measured immediately upon delivery into target cell cytoplasms, where the pH is neutral. This is followed by cleavage of procaspase 3 by granzyme B in the target cell cytoplasm and the subsequent induction of apoptosis. All proteolytic events can be quantitated at the live single-cell level by flow cytometry and imaged by confocal microscopy.
I understand very little of what you just said. Maybe we should talk more about this.
I just need to know if 3 patients are enough. My grant is due tomorrow.
We will need to meet to better understand this before we can determine the appropriate sample size.
I can put you on my grant for 0.5% FTE. You can design and analyze the data later.
Do you really think that is enough to do the work?
Well, it may be less if it is awarded and the budget is cut.
Please go away.
Now, I know that none of the people on this call would ever tell you to go away. But all of the statistical collaborators that you will come into contact with during the course of your career have had some sort of conversation that sounded a lot like that. And so my hope for today is that we will give you what you need to know so that you don't go into a meeting to start a talk about how big your trial should be and what things need to be considered. That turns into the disaster that we just witnessed. Okay, so let me now share my actual slides. Okay, share this. You see my slides?
Yes.
Yes.
Awesome. Thank you. Perfect. There is a link to that video and another in the slide deck, which I'm sure will be made available to you. But I'll start this talk just by some very brief disclosures. I am a faculty member within a Department of Public Health Sciences, and my department does receive some consultant fees related to my service on a data and safety monitoring board for Alnylam. But I'm not going to talk about any unlabeled use of any commercial products or really even any investigational use in this talk. I usually start this talk with a disclaimer because in reality, I could teach a whole semester-long course on clinical trial design and not be able to cover everything that is available and address every question that you could think of. So in order to do this and give you a reasonable scope within an hour, I need to keep at least the beginning part of this talk focused on a specific framework. And the framework that we're going to be operating under is that of a superiority trial design. And what I mean by that is a two-arm randomized trial where you really want to show that one treatment is better than the other with regard to a primary outcome. And where the deliverable of your research is the p-value from a hypothesis test. Now you may say, but Sharon, I'm only in the phase 2 space, so this isn't really relevant. The primary outcome that we're talking about doesn't have to be the clinically relevant outcome that you might use in phase 3. It could be a biomarker outcome. It could be a surrogate endpoint. It could be some sort of feasibility measure. Okay, but just to keep it sort of the scope limited, we're going to think about a two-arm randomized trial. There's lots of other designs where you might not be interested in demonstrating superiority. Maybe you're interested in futility or non-inferiority, or where at the end of the day, you don't really want to do a hypothesis test. You want to use selection theory to pick the treatment that's going to move forward, or just come up with an estimate of precision associated with some measure. And in those cases, some of the factors that we're going to talk about in the first part of this lecture are going to be more or less or not at all relevant, okay? So to start off with, I want to talk about this idea that clinical trials are really models, and those models require that we make tons of assumptions, okay? We make assumptions about dosing, right? We assume that the dosing we think we know from animal studies is relevant to the humans we're about to conduct research on. We assume things about the duration, that the duration that was efficacious in animal studies is actually feasible when we talk about human trials. We make assumptions about outcomes and whether the effect size is going to be reasonable, whether the effect size is going to be large enough to allow us to detect it with a reasonable sample size, and that any noise that's in that outcome can be understood either with protocol standardization or other such things and overcome by the treatment effect. We also assume that subgroups respond equally to the intervention, right? At the end of the day, what that means is there's lots of things that we're assuming, and we have to understand the big search space for all of these questions. And most trials are then making lots and lots of compromises to reduce the number of those parameters in order to make the model solvable. That is to make the trial something that we can do in a reasonable time with a reasonable number of patients and resources. So the goal, as I think about it, and I can't take credit for this verbiage, this is one of our Siren collaborators who talks about the idea that you can conduct a postmortem of your trial before you start the trial and think about all of the things that could go wrong, what the end impact might be on the results, and then try to fix those things up front. So at the end of the day, you don't have those regrets. Okay. So in other words, if you're not sure about the dose, maybe you shouldn't be testing just one dose. Maybe you should be doing a dose finding study, right? So thinking about those things on the front end to minimize regrets. And not necessarily a secondary goal, but maybe a co-goal is to conduct a clinical trial, regardless of what space you're in, such that the clinical community and the funders know what to do next, right? That the question has either been answered in such a way that now you know what to do, or the question has been answered in such a way that the next step is obvious, okay? These are not as easy as it seems, but it's really important for us to go through this process because coming up with the sample size that is exactly right does a number of things for us. One, it provides assurance that the trial has a reasonable possibility of being conclusive, right? If you're going to conduct a study with only a 20% chance of showing the difference that's meaningful, why do the study? It also allows us to really focus on exactly the number of patients that we need so that those resources are efficiently allocated to your trial and to all of the other hundreds of research questions that are on the table at the funding institute at any given point in time, right? There's also ethical issues associated with doing a trial that is either too big or too small. If the trial is too large and you could have answered the question with half the patients, then effectively you've withheld a potentially efficacious treatment to the entire second half of your trial population, right? And we certainly don't want that. If the trial is too small, then you are exposing patients to any potential risk that's associated with your intervention without the ability to really answer the question of interest. And that's not really fair to the participants either. So again, we're trying to sort of find this sweet spot. And I'm going to show you two examples of why it's important to be very thoughtful about how you're going through this process. But I'm sure that there are lots of other examples in all of the fields of study that you all are most interested in. I'm going to start with one that I became familiar with, I don't know, it's probably 8 years or so ago now, when we were designing the pediatric ICE-CAP study. So they conducted a study of children with out-of-hospital cardiac arrest, and they randomized patients to 120 hours of either therapeutic hypothermia or therapeutic normothermia. And they followed those patients for 12 months to evaluate the primary endpoint, which was defined as survival with a good neurobehavioral outcome. The sample size was just under 300 patients, which for a definitive study sounds small, but could be reasonable. And they said that that sample size left them powered to detect an absolute effect size of 15 to 20%. And the results were published in the New England Journal in 2015, and they showed that while only 12% of the normothermia arm achieved that good outcome, 20% of the hypothermia arm achieved that good outcome. Now you can see that's an absolute effect size of 8%, which is about half what they were powered for. And unsurprisingly, the p-value was not statistically significant. So the conclusions in the manuscript are that therapeutic hypothermia did not confer a significant benefit in survival with a good functional outcome. But as one of the limitations, they have to address the fact that they can't rule out a clinically important benefit that is smaller than the one that they were powered for. And in this, perhaps in lots of disease spaces, but particularly in this disease space where we're talking about cardiac arrest in children, An 8% absolute improvement in survival with a good outcome seems like it might be something that is clinically meaningful. Another example is a more recently completed trial in adults with severe traumatic brain injury. These patients were randomized to management by either ICP monitoring only or both ICP and PbTO2 monitoring. And they were followed for 6 months for a binary outcome based on the Glasgow Outcome Scale Extended. And that binary outcome is effectively poor outcome. And so they were powered to detect a relative reduction in poor outcome of 30%, which is an absolute reduction of 17%. And they said they needed 300 patients. So the results were published in Lancet Neurology a couple of years ago. And in the top half of this graphic, you can see the distribution of the Glasgow Outcome Scale score at 6 months, and the bottom panel is at 12 months. And let me see, I hope you can see my mouse. These lines that connect the results for the combined arm versus the ICP only arm are effectively showing you what the treatment effect looks like at each of these potential binary cutoffs. And so 1 through 4 would be about here. Okay, so you can see at 6 months, there's not much of a difference, but at 12 months, there is a much larger difference. Okay, so again, a reason to be really thoughtful about what time points you're choosing, what effect sizes you're targeting, what patient populations you're studying. It seems very silly, but these are all very important decisions that really have impact on the conclusions we're making. Okay, so first things first, for the first part of this talk, we're going to talk about some statistical concepts. And the first of those is, how do you state the research question? Many of you have already given elevator pitches on your research topics, and I'm sure that you can tell me in 3 minutes or 5 minutes or 10 minutes what the question you're interested in addressing is. But when you start to think about establishing a collaboration with a statistician, that 3 or 5 or 10-minute elevator pitch needs to be boiled down to something that is a testable hypothesis, okay? And your statistical collaborator can help you with that. But what that means is we need to be really specific about what we're looking at. What's the patient population of interest? What's the response variable that you're looking at? And at what time point are you measuring it? And what are you comparing it to? Is it a parallel group design? Is it a pre-post design? All of this is important because it informs how your statistical collaborator is going to start thinking about what that design might look like, what that analysis might look like, and therefore what the sample size might look like. So the key to stating statistical hypotheses is really that we assume that there are 2 states of nature, and those 2 states of nature are reflected by 2 hypotheses. The easiest way to do this, I think, is to start with the alternative hypothesis. Okay, that's the statement you want to be able to conclude. So when you and I meet and you tell me about your research project, you say, I think treatment X impacts outcome Y, right? That's your alternative hypothesis. The null hypothesis is the opposite of that. If treatment X doesn't impact outcome Y, then there's nothing going on. That's your null. These 2 hypotheses have to be mutually exclusive. Okay, that means either one is true or the other is true. They can't both be true. And they have to be comprehensive. So there's not some third option hiding out in the background that could explain what's going on. Okay, so that's important. Because what we're really doing when we conduct a statistical hypothesis test is to establish proof by contradiction. So we conceptualize nature as being in one state or the other. As statisticians, certainly, as scientists, often, what we are comfortable with is assuming the simpler state, right? So we start in the frequentist framework by assuming that the null hypothesis is true, nothing is going on. And we know what that should look like, right? We know what the variability in our outcome looks like, that sort of thing. So we know what we would expect if nothing is going on. And then we collect data from the real world And if our data clearly contradict that null, then we have to scrap our belief in the null and say that the alternative must be true. But if what we observe is consistent with the null, or at least not uncommon under what we would expect from the null, then there's no reason to go for that more complicated explanation. We can keep our belief that nothing is going on. Okay, so we've learned that there are two possible states of nature, and those are captured with two statistical hypotheses. Okay, the null hypothesis is the simpler one. The alternative is the one that you want to show. There's only two possible decisions that we can make as the result of a statistical test. And this is really important, and it's something that I often see misstated in the literature. So the only possible decisions are reject the null and conclude that the alternative works, or fail to reject the null. Okay, you can't accept the null. We don't say we conclude the null. We can either reject it or we can fail to reject it. So if we think about those two true states of nature that are the columns, either the null hypothesis is true or it's false, and the two decisions, which are the rows, that leaves us with four possible outcomes. Two of these are correct decisions, and two of them are mistakes that we want to avoid. Okay, so we'll start with correct decisions. The first correct decision is here, where we reject the null hypothesis when it's false. Okay, this is fabulous. You get a significant test result from your statistical analysis. You write up a paper, you attend a fantastic conference, and you start building this research portfolio based on this finding, right? It's wonderful. There's another correct decision, which is slightly less exciting, but is still a correct decision, which is that the null hypothesis is true and you fail to reject the null hypothesis, right? That's less exciting for lots of reasons, but it is still a correct decision. And so we want to go about our research plans or our research development in such a way that we are maximizing the likelihood that we fall in these two categories, or limiting the likelihood that we fall in the categories that reflect errors. Okay, so let's talk about those errors. The first error is the scenario where the statistical test leads us to reject the null hypothesis, even though the null hypothesis is true. Okay, this is effectively a false positive. You're concluding that the intervention works when it doesn't. And there are consequences associated with that, and the consequences depend on what the intervention is, what the outcome is, what the population is. But it's important to be aware of that potential error and what its consequences are. The other possible error is that the statistical test leads us to fail to reject the null hypothesis when in fact it's false. So the intervention does work, but you are unable to claim that it works. Okay, so that's a false negative. We go about our statistical tests by, again, setting things up so that we are managing the likelihood that we wind up in either of these error spaces. Okay. Okay, so why are we talking about the statistical analysis? That's because it's an important factor in determining the sample size. In order to have a conversation that gets you to the number of patients you need for your particular trial, we need to talk about and understand what the likelihood is of a false positive. How likely or how willing are you to make that mistake? We also think about how likely do you want it to be that you will find an effect when it exists? Okay, that's the power. Those are the two key components. But in order to establish how the power relates to the sample size, under those conditions, we also need to understand what the impact of the minimum scientifically important difference is, what the variability in your response is, and what the experimental design is that you're focusing on. So we'll start by talking just a minute about the level of significance. So I mentioned this earlier, it's the false positive rate. Many of you have seen level of significance reported in the literature as 0.05. I would be surprised if you saw it very often at anything other than 0.05. But 0.05 is not a magical number. Okay, so how you set the level of significance should really be based on the interpretation and the consequences and your willingness to make that mistake. So the interpretation of the level of significance is that even when the null is true, so even when your intervention doesn't work, you're going to conclude that it does alpha times 100% of the time. Okay. Now, the reason that this is important is because what I'm showing you here is the distribution, say, of a test statistic under the null hypothesis. Okay, so under the null, where your intervention does nothing, the test statistic is going to be centered at 0, but there is some reasonable likelihood that you will see test statistics all the way out to greater than -2 or greater than +2, right? Even under the null hypothesis. What the level of significance is doing is translating what we know about our willingness to make that error and using it to tell us how far away from the center point of this distribution do we have to be before we're willing to say, nah, that's not likely. We're going to believe the alternative. Okay, so what I've shown here is the shaded region for an alpha, a one-sided alpha of 0.1. Okay, so if you observed a test statistic out here in the shaded region, you would say that's not very likely under the null hypothesis. I'm going to go with the alternative. Okay, if I reduce that level of significance to 0.05, my shaded region gets smaller, my rejection region gets smaller. I make it harder to declare a win. Okay, and we can reduce it even further to 0.01, right? And again, everything gets smaller. So that is going to have an impact on the sample size that you need to conduct a certain analysis. Okay, now we'll talk about the power. We talked a little bit about the error that's associated with the power, but while we specifically set alpha, the false positive error rate, we don't usually think about setting beta, the false negative error rate. We set it inadvertently by setting a target power for our trial that is high. Okay, so the power is the probability that a test is going to reject the null hypothesis when in fact it's false. The probability that you're going to declare your intervention works when it actually does. So the interpretation here is similar, but a little bit different in an important way. So if your power, let's say, is 80%, the interpretation is that for a given value under the alternative hypothesis, we're going to reject the null 80% of the time. Okay? For a given value under the alternative hypothesis. That's really important. That should tell you I have to set what that value is for starters that I want to power for. And it should lead you to thinking critically about papers that you might read or grants that you might review that say, I have such and such power for this trial without giving you those specifics about what treatment effect it is actually powered for. Okay, and this is the relationship with sample size that everybody is sort of focused on. But again, it incorporates all of these other components. So here's an example. This is sort of a generic example where along the x-axis, we have the total sample size. Along the y-axis, we have the power. And everything else held constant. So all of the things we haven't yet covered, fixed. You can see, just as I said, as you decrease your alpha level, the power associated with your test decreases as well. Okay, so again, it's important to keep that in mind. Now, I harped a little bit on the fact that the power is specific to a given effect size. And the effect size that I would encourage you to be thinking about before you have this initial consultation is the minimum scientifically important difference. Now, that's the smallest difference that would mandate a change in scientific practice or understanding, assuming that there's no serious side effects or excessive costs or something like that that would lead you to want to favor one over the other. Okay. In some areas, in some clinical areas, the minimum scientifically important difference is either clearly known or clearly assumed. Okay, so for a long time in stroke trials, we said 10% on a binary MRS outcome. That's what we were designing trials for. In some cases, that may not be the case. You may have to get together a group of experts and talk about how big would this effect have to be in order for me to go through the effort of procuring whatever machinery or equipment is required and that sort of thing. But the reason that we focus on this is because of how tightly changes in that difference are tied to the number of subjects that we need. So a well-known rule of thumb is that the larger the difference you want to detect, the smaller the sample size you need to detect it. It's a little bit simplistic. I'll show you in a minute, but for the purposes of this talk and that initial meeting with your statistical collaborator, it's a good place to start. So here, again, this is a generic power curve where the sample size is along the x-axis, power is along the y-axis, and again, holding everything else constant, fixing the level of significance at whatever value it is, fixing the variability in the design. As you decrease the difference that you're trying to detect between the two means, the power associated with a given sample size goes down. Okay. Now I said that rule of thumb is a little bit simplistic, and it is because it ignores the contribution of variability. And what we mean by variability depends on what kind of outcome you're talking about. In the continuous case, it's your usual assumption of variability, right? You need to know what the standard deviation or the variance of your endpoint is. Again, here, a standard power curve, and you can see that everything else hold constant as the variation or the standard deviation for your particular endpoint decreases, the power associated with a given sample size is going to increase. Okay, so anything you can do in terms of clinical standardization or measurement standardization, those sorts of things are going to be helpful in ensuring that you have as much power as you can get. If your endpoint is binary, what we mean by some indication as to the variability is really an estimate of the control rate. Okay, so if good outcome is what you're looking at, how likely is it that a control member is going to have a good outcome? And again, you can see here that as the control proportion, or what's labeled here as the reference proportion, changes within a given sample size, the power is affected. The power for a binary endpoint is such that as the control proportion approaches 50%, the power is going to be reduced. So as you move from the end of the proportions range towards the middle, the power is going to be reduced, and you're going to need more subjects to overcome that noise. Okay, so we talked about the statistical stuff that you'll have to keep in mind, but maybe even more important than that, when you are thinking about how to go about this process of deciding how big a trial you need, are to think about all of the ways that the experimental design can make your trial more efficient or could be a limitation of the efficiency that you can achieve. So I'll go through some examples of some of these questions, but I think it's important to really consider, especially in the earlier phase trial space, right? Is a parallel concurrent control group really the best way to answer your question? Okay, it might be. I'm not saying it never is. But if it is, then be thinking very carefully about what that control group actually reflects. And think about whether, again, if it's not, maybe there was a trial that was just completed in your disease area using your outcome and a very similar patient population, and maybe that has historical data that you could use to help give you a little boost, right? Think about whether a hypothesis test is really the best way to answer your question. Again, it might be, but in some cases, trying to go about answering a question with a hypothesis test may require more patience than, say, trying to answer your question with selection theory. Okay, if you're trying to evaluate 4 different treatments and trying to decide which one of those should move forward for further testing, comparing all of those 4 with a hypothesis test is probably going to cost you more in terms of subjects than using selection theory to say, I want to be certain that the best one is the one I pick to move forward. Okay. If you think a hypothesis test really is the way to go, be thoughtful about what sort of hypothesis you're testing. Again, I mentioned that we're doing this in the superiority framework for the first half of this talk, but you could reframe a phrase 2 question as a futility hypothesis and perhaps give yourself better statistical standing, still with a reasonable sample size. Think about how confident you are in the minimum clinically important difference so that you're not in a situation like THAPCA where we're powered for 15 to 20%, but we observed 8%. Think about whether you can answer multiple questions in the same design. Now, you're probably not going to be able to evaluate 15 doses and 15 durations in the same design, but if you have 2 relatively simple questions, can you merge those into a single design and be able to answer both of them with the same patient pool? Think about whether an adaptive design is really needed. I don't know if Dr. Moyer is on the phone. He may be surprised to hear me ask this question. Sometimes it is needed, sometimes it's not. And you want to be thoughtful about what those adaptations are. And we'll talk about why that's important. And the reason that all of this matters is because the sample size calculation depends on the method of analysis, which depends on the design. So you can't just come and say, this is what I want to study. How many patients do I need? Like the people in our video. We need to know all of this background information in order to make sure that we're giving you something that makes sense. Okay, so these are real world examples from trials that we have done. I probably should update some of them within the last couple of years. But this is an example of evaluating what is the control group and can you answer multiple questions in the same trial. Okay, so we were working on a trial design that initially said we're going to take 6 treatment arms and we're trying to look at which of these is better for this particular patient population. Okay, but as we're having this conversation in grant development, it became clear that although no reversal treatments to me as a statistician sounds like a control arm, that's not really the standard of care if we're thinking about what sites are commonly doing in this patient population, right? So common practice may not be doing nothing. Common practice may be one of these other arms, and guideline-driven care may be a different arm. So really thinking about if we're going to do this as 6 different arms, what are we comparing to? Because no reversal treatments may not be the right comparison. What do we do if there's variation across sites in standard of care? How do we accommodate that when we're making this comparison? And if that's true and there is variation across sites in standard of care, do all of the sites have the same level of equipoise to randomize to all the arms? Maybe not, which adds another layer of complexity. But in terms of how do we select which of these or evaluate what the effect of these treatments is, it may be better to think about this not as 6 independent arms, but as a 3x2 factorial, where all of the patients can contribute to what we can say about all of the treatments. Okay, so that's one example. Here's another example where being a little more creative, a little more flexible in how you specify your hypothesis can be helpful in terms of giving you a solid statistical standing. So this is a phase 2 clinical trial in subjects with acute ICH. A common complaint about phase 2 trials is that they often look like underpowered phase 3 clinical trials. And in this case, that's where we started with the discussion. So our usual alternative under the superiority framework would be that the difference is not 0, Typical alpha would be 0.05. We know from past studies that the reference proportion is 56%. And we want to see if there's an effect size of 12%. And I'll tell you, 12% is bigger than we would have used in a phase 3, but we were trying to get a sample size that was reasonable. And 310 patients in this phase 2 trial only gave us 59% power under this framework. That's not great. But if we flip how we think about it and we say, well, gosh, in the phase 2 space, I don't really want to show convincing evidence that it works. I want to know whether I should abandon it or whether I should move it forward, right? So if we flip that alternative hypothesis and say, no, what I really want to show is that the treatment effect is less than some clinically meaningful threshold, right? In this case, 12%. That helps in a couple of different ways. One, my alternative hypothesis is now one-sided, which streamlines things a little bit. And my alpha level, I can be a little more flexible with because the consequences of making a false positive declaration in these two cases are very different, right? In the superiority case, if I make a type 1 error, I'm saying an intervention that doesn't work does work, and I really want to minimize that likelihood. But in the futility case, a false positive means I have an intervention that works, and I decided not to investigate it further. I don't want to be too willing to make that mistake, but 10% might be okay, right? So again, under this sort of framework, I can flip this around, and now 310 patients gives me 80% power to test this hypothesis, and that's a solid go, no-go decision in the phase 2 space. Okay, I mentioned adaptive designs, and I'm going to go through an example really quickly. I guess the first thing I want to say about adaptive designs is that it is important to understand that adaptive designs are not reactive designs. Adaptive designs means you have to carefully think through all of the ways that things could happen in your trial and pre-specify how you want the trial to adapt in those scenarios. And so there is not generally a closed form solution for this. There's going to be a lot of back and forth between the clinical colleagues and the statistical colleagues, but they can be very helpful in certain spaces. So at the beginning of the adaptive process, you're going to start by enrolling some patients, getting some data, analyze that data, and then evaluate that data against whatever pre-specified stopping rules or adaptation rules you've specified. And then adapt in, again, whatever way you have pre-specified, and this can continue going. So here's an example that we worked on a couple of years ago. We were randomizing eligible subjects to placebo or 4 active doses and following them for a primary outcome of just 24 hours. And after a brief burn-in period where patients were it wasn't exactly equally assigned, but that's what I'm going to say because I can't remember what the exact proportions were. We're going to update the allocation probabilities so that we put more patients on the doses that look like the ones that we're trying to identify, right? I mentioned already, these designs generally don't have closed form solutions, and so we determine the operating characteristics, the type 1 error and the power by simulation. So we simulate a trial under the null, meaning nothing's happening, under the alternative, potentially lots of alternatives, and then we track how each of those trials did and what decision they made, how many times they adapted, and so forth. And we really are trying to stress test the design that we've built under different assumed truths and make sure that any single trial behaves in a way that makes us feel comfortable as investigators, but also to understand the operating characteristics by summarizing across a large number of simulations. So at the end of the day, this is a lot to say that there is a lot that can go into that conversation. So you probably should not expect to have one conversation and get a sample size at the end of it. There's going to be back and forth. There's going to be questions where the statistician says, I don't understand this. Can you help me understand the outcome or the effect size or whatever? But at the end of the day, you will come to a place where you have a sample size and you have to be able to justify it. And it's very important that you do this carefully. It can be very important during grant review. So you want to say, how many subjects are you requesting, whether or not that sample size is adequate. And you want to be honest about that because the statistical reviewer will be able to tell if you're not. So if you need more, that's okay, but tell us why you can't do more and what you can do with what you have. And if you're actually asking for more than you need for the primary outcome, that's also okay. But again, explain why. And in order to do that and to make folks feel comfortable that you really have the number that you need, you're going to have to be able to address all of these questions, right? So what was the difference? What was the power? What's the type 1 error? What design are you using? What's the analytic plan? All of that has to be there in order for the justification to come across as effective. There are a couple of logistical things that I'll just mention that also impact the sample size, although we often spend a lot of time dealing with the stuff I've just talked about. Less time sometimes with the logistical impacts of anticipated recruitment can be really important for adaptive designs. If you've specified an adaptive design, but all of your patients are going to be randomized in the first month, you may not have any data to adapt according to. Treatment crossovers have an impact on the treatment effect that you're estimating. It often dilutes that treatment effect. And so you're going to want to likely bump the sample size in order to account for that. Protocol non-adherence can do the same thing. Loss to follow-up and consent withdrawal. You're going to want to think about whether your analysis is going to include those patients who were lost and didn't have the primary outcome observed, and how those are going to be handled and what the impact is going to be. And the interim analysis, if you're pre-specifying any sort of group sequential spending, the operating characteristics can be impacted as well. And so we typically inflate the sample size to accommodate those. There is freeware on the web. You know what? I have done this talk so many times that I didn't actually test to see if those links were still available. So I will check and I will let Megan know if they are not and they need to be corrected. There are also software available for purchase. Inquiry is one of them. Pass is another. Your statistical collaborators are going to use a wide range of things from SAS and R to EAST. And some of the freely available software. And then I will end with this quote from Russell Lenth, who was a professor at the University of Iowa. And he, at some point, had on the web this story that says, I received quite a few questions that start with something like this. I'm not much of a stats person, but I tried XYZ. Am I doing it right? And he said, please compare this with, I don't know much about heart surgery, but my wife is suffering from XYZ and I plan to operate. Can you advise me? Like I said, there's lots of software available. You can plug numbers into any of it, but that doesn't necessarily mean that what you get out will be relevant to your particular scenario. So I think the best advice that I could give you would be to establish a relationship with a statistical collaborator. So not just sort of finding someone, can you help me with this project? Can you help me with this project? But at your stage of the career, it can often be mutually beneficial for you to find a statistical collaborator who is at a similar stage in their career, who has an interest in your type of research, and then you can do work together that will benefit both your areas of research interest and theirs. So just something to think about. Okay, stop sharing and see if there are questions.
Thank you, Sharon. That was excellent. We do have one question in the chat. Others are welcome to put questions in the chat, or we can do hands raised after this one. So it says, thank you for this great talk. What influences whether a statistical test should be two versus one-sided?
Yeah, that's a great question. So I really think that it depends on the context of your statistical test. So if you are trying to evaluate, say, an adverse event endpoint, and the comparison group is a control or a standard of care arm, you're probably not expecting to show that the intervention actually has a reduced side effect profile, right? Some adverse events are going to happen even in the standard control arm. So maybe there you really are only interested if the intervention increases that whatever adverse event that you're looking at. It can depend on the stage of the study that you're looking at. Again, thinking about whether you would be interested in finishing the trial to show whether something is worse than doing nothing is important. But in many cases, even when you make it one-sided, we split the alpha level. So you can, if you can sort of cheat the system a little bit, if you go from a two-sided test to a one-sided test, but you don't split the alpha level. So in most scenarios, we would say, okay, if you want to do a one-sided test, you can do it, but you're going to do it at one-sided 0.025, which means the rejection region would be the same as if you were to do a two-sided test.
Thank you.
Are there other questions? Thanks, Robin. Okay, well, if anything comes up after, feel free to shoot me an email. I am happy to talk by email or offline at any time.
Thank you so much, Sharon. Thank you, everyone, for joining us. Please remember to grab that survey link in the chat. Our next webinar is coming up in just 1 week on May 15th and will be presented by Dr. Lara Gihai from the Cleveland Clinic, and her talk is Computational Approaches to Optimize Clinical Trial Recruitment and Conduct. We're really excited to have Dr. Gihai present a new topic in our series about ways to use emerging technologies. So we hope to see you there. Have a great Friday and a great weekend.
Thanks. Bye all.